This video will cover how to gram stain bacteria. The gram stain is the most important staining procedure in microbiology. It is used to differentiate between gram-positive organisms and gram-negative organisms. Hence, it is a differential stain. Now, gram staining involves a four-part process which includes crystal violet, the primary stain, iodine acting as the mordant, a decolorizer of acetone or some alcohol, we will be using ethanol, and safranin, the counterstain. Now, gram-negative and gram-positive organisms are distinguished from each other by differences in their cell wall, and these differences affect many of the aspects of the cell, including the way that the cell takes up and retains stains. Gram-positive cells take up the crystal violet, which is then fixed in the cell with the iodine mordant. This will form a crystal violet iodine complex, which remains in the cell even after it's decolorized. It's thought that this would happen because of the cell wall of gram-positive organisms, including a very thick layer of protein sugar complexes called peptidoglycan. This layer makes up 60 to 90% of the gram-positive cell wall. Decolorizing the cell with the ethanol causes this thick cell wall to dehydrate and to shrink, which closes the pores in the cell wall and prevents the stain from exiting the cell. At the end of the gram stain procedure, gram-positive cells will typically stain a purplish blue color. The gram-negative cells, on the other hand, take up crystal violet and iodine and form a crystal violet iodine complex in the cell, just like the gram-positive cells. And so, at our first couple of steps, you will not see a difference between our gram-positive and our gram-negative cells. However, the decolorizing step with the ethanol causes the cell walls of the gram-negative organism to not retain this complex. And this is believed to be possible because the peptidoglycans are present in the cell wall of gram-negative organisms at only about a 10 to 20% ratio of the cell wall. And so the gram-negative cells would also have an outer cell wall that the gram-positives do not have. And this layer is made up of lipids and polysaccharides and proteins, and exposing the gram-negative cells to this decolorizing dissolves the lipids in the cell wall, and it allows the crystal violet iodine complex to leak out of the cell. And this allows the cells to stain with the counterstain safranin, so at the end of the procedure, our gram-negative cells will appear a reddish pink. And so here we have a representation of both the gram-positive and the gram-negative cell walls. Now, our gram-positive have a thick peptidoglycan layer, where our gram-negative have a very thin peptidoglycan layer. It's this thick layer that allows the crystal violet iodine complex to hold tight to our gram-positive bacteria, giving them that purple blue color. And that thin peptidoglycan layer, when we have that decolorizing step, this is where we lose that crystal violet iodine. And then when we add the safranin, that counterstain, that's when this cell will pick up the counterstain and will show a pinkish color. So step one in preparing a gram stain is to heat fix your bacteria to the slide. And if you need to, refer to my previous video on heat fixing. But heat fix your bacteria. We will be adding the various stains in the following order. We will first be adding crystal violet. This is our primary stain, followed by iodine as our mordant, followed by our alcohol, our ethanol, as our decolorizer, and then finally as our counterstain, we will be using safranin. So step one in our gram stain, after we have heat fixed our bacteria, we will saturate. Now I know that my directions up here says a few drops, but let's saturate this bacteria smear with crystal violet and let the crystal violet set for 1 minute. Now if you go a little longer, that's fine, but at the end of 1 minute we will rinse the crystal violet off. And here we are rinsing the excess stain off with water. Now note that we are holding this at an angle and that our bacteria smear is right in this area, and that we wash with the water hitting above the bacteria smear. This way we will not wash the smear off, and as the water drains down it will wash the excess stain off while the smear stays intact. We will then blot dry our slide before we add our next stain. Okay, our second stain that we add is iodine, and again I know our directions say add a few drops, but I like to saturate our slide with our iodine, and this right here will act as our mordant, which will cause our crystal violet complex to latch onto the peptidoglycan layer. So we will let this sit roughly a minute. And you'll notice that our steps require stain, rinse, blot, stain, rinse, blot. We see this a lot. But we have our smear right here, and again we are going to rinse with water, rinsing above the smear, holding this at an angle so the excess stain will drain off. We will then blot dry, gently blot dry our slide with a paper towel so as not to remove the bacteria culture. Holding our slide at an angle, we will add our ethanol, and we add our ethanol for roughly 10 seconds. I just say count to 10 slowly. We will add the ethanol up above, and we will let it drain down, and it will drain right into the sink. Once we have completed this, again we will rinse and blot dry our slide. At the end of rinsing and blot drying, we will then add our counterstain. We will let our counterstain set for 1 minute. This is the safranin. At the end of the 1 minute, we will again rinse the slide, letting the water hit above, draining down, blot dry our slide. Here we are with that last step where we're rinsing off that counterstain of safranin. Again, we are doing that above our bacterial culture, so we're letting our stain drain down. We blot dry it. So at the end of a gram stain, your slide will either look like this one right here on the left or this one right here on the right. And so this, it appears that we have a gram positive purple, and here we have a gram negative pink. Now, it's rare that it will be this cut and dry, and many times you will have to look under the microscope to see what your cells look like. And so here we have our gram-positive bacteria, and we can see we have some rod-shaped and chains, so it looks like we have a streptobacillus here. And here we have our gram-negative, and it looks like we've got some staphylococcus going on over here. But our gram-positive will be purple, and our gram-negative will be pink. Now, I suggest that the very first thing that you do is learn to master a gram stain, and anytime you have to identify an unknown bacteria, the very first test you want to run is a gram stain. This will differentiate your bacteria as either gram positive or gram negative, and in essence, that long list of unknowns that your bacteria can possibly be will be cut in half. And once you know what you're working with, you can then plan out your next series of tests to identify that unknown.